Students, welcome back. And today we are going to be looking at things very, very closely. Very closely. Yes, we're going to be using the microscope today to complete our lab activity. But before we get to that, let's just tie up some loose ends. Let's just finish our notes from yesterday, which actually is a great transition into today's activity. So open your books if you haven't done so already, and let us pick up where we left off yesterday when we were discussing typical plant and animal cells. You may recall these diagrams. Great pictures here of a typical plant cell, and you can see the various organelles which we've been discussing this week. Notice some of the more notable things regarding a plant cell, such as the cell wall, and all of these green chloroplasts that are only found in plant cells and not in, not found in animal cells. You don't see chloroplasts and cell walls in animal cells, as cool as that might be. But these are pretty detailed diagrams. And today in the lab, we're going to be comparing plant cells and animal cells and distinguishing between the two, trying to determine what kind of a cell it is based on our observations under magnification. So before we get to that, let's just use some simpler diagrams here of an animal cell and a plant cell. You should see this in your book. May look a little bit familiar to you if your books are open. And in your book, you can probably see these labels a little bit more clearly as well. And what I'd like to do is take a few moments now for you to create a Venn diagram. I know you're all familiar with Venn diagrams, but in case you're not, let me just explain it briefly to you. Venn diagrams are very useful tools when comparing and contrasting two or more different things. In this case, we are comparing animal cells and plant cells. And so what we have here in this Venn diagram are two circles that are joined in the middle. In the first circle, which is underneath the plant cell, you're going to be listing some things that are unique among plant cells. And under the diagram of the animal cell, you're going to list some things that are unique about plant cells. So the two outer portions of the circles would be the contrast, the differences between the two. The part where the two circles join, however, this is your opportunity to list things where there are similarities between both plant and animal cells. So I'm going to give you a few moments now to discuss and write down what you think might be appropriate to complete your Venn diagram. So pause now and continue. Okay, hopefully you've had enough time to talk about it. And there's no one right answer here, but I am going to give you one of several possible correct answers as I complete my Venn diagram. Animal cells, as you know, they lack cell walls and they lack chloroplasts, whereas plant cells have both cell walls and chloroplasts. So those are differences. That's how you can contrast the two. Now, how can you compare them with similarities? Well, they're both basic building blocks. They're both cells. Even though one is for plants, one is for animals, they are both still basic building blocks of living things. And they do share other organelles as well. So this is one of several possible Venn diagrams that you could have illustrated. Now, incidentally, creating this Venn diagram will help us when we complete our lab activity. And as we think about our lab activity, let's turn to it now in our books. And the question of today's lab is, how can plant and animal cells be told apart? Now, that doesn't mean you're going to tell plant cells, animal cells, divide yourselves, stay apart from one another, wear your masks. No. That's not what it means. What it means is, how can you tell the difference between a plant cell and an animal cell in the lab? How will you be able to tell the difference between the two? I'd like you to take a moment now and write a hypothesis and then share your hypotheses. Share your thoughts with one another while you pause the video. All right, we're back once again, and hopefully some of you may have mentioned some of those things you may have written about in your Venn diagram, the similarities and the differences between plant cells and animal cells. You've got to know how they compare and contrast with one another if you are to be able to tell them apart under magnification, which now brings us to our lab activity. So we're going to do this together. And so let's transition over to the lab now, shall we? So here we go. Whoop, peekaboo. All right, here we go. Whoop, peekaboo. Here we go. We're in the lab. Can you see me? Can you see me now? Can you see me now? All right, we're going to go through the various steps of this particular lab activity. The first thing that you need to do is acquire a microscope. Raise your hand if you remember the proper way of transporting a microscope. I hope you remember the ABCs. The arm, the base, and carry it close. I said the arm, the base, and carry it close. Everybody, the arm, the base, and carry it close. You say the arm, the base, and carry it close. That's right. It's a catchy little rap, don't you think? Anyway, once you have obtained your microscope at your table, you are to remove the cover, which keeps the dust, dirt, and grime and the germs off of the microscope itself. And just a little refresher here on the microscope. I am going to point to a part of the microscope, and I'd like you all to yell out what is the identity of that part of the microscope. Are we ready? So I want to hear you loud and proud as I point to the various parts of the microscope. So here we go. I think we got them all. Eyepiece lens, arm, base, body tube, nose piece, objective lenses, coarse adjustment, fine adjustment knobs, the stage, the light, and the diaphragm. So that's a good review on the parts of the microscope. Now, once you've obtained the microscope, I want you to remember some things here when beginning the use of your microscope. Which objective lens should we all have pointing down? The low power objective lens is always pointing down, and hopefully you remember why. Next, we use the course adjustment knob to raise the body tube all the way, and then we're ready to place our specimen found on our slide on the stage for us to observe. By the way, anybody remember what these are called? The stage clips. The stage clips. Now, today we are going to be comparing typical plant cells and animal cells. We're going to be comparing and contrasting the two, and you are going to have to identify which one is which, and hopefully be able to see some organelles in the process. Before we get to the actual lab, however, or your observations of it, I should say, let me put these off to the side so you can't see which one I'm taking. I just want to go over with you how to place the slide on the microscope. Anybody remember how we should obtain a slide? We should get the slide by the side and hold it by your side. Very good. Should we ever grab onto the top and bottom of a slide? Please say no. But rather you get the slide by the side so your grubby fingerprints don't smear and smudge the specimen that we're looking for. Many of the slides we'll be using actually have labels on them. I'm not going to show you this label because then I'll give you the answer. But you can hold on to the side of the slide with the label on it. That would be great because then you're not smudging anything that you need to see. Once you've obtained the slide, you examine it first with your eye. Oftentimes you will be able to see a specimen under the cover slip of the slide. And ideally what you need to do is place that specimen in the center of the diaphragm, the opening of the stage. Once you've positioned it in the center, you can actually use the stage clips to position it in a way that it won't slide around the stage. Now it is held into position. So far, so good. Next step, while you're looking at it from the side, making sure you have it on the low objective power lens, use the course adjustment knob to lower the body tube all the way down. There's plenty of room. You're not going to smash the glass slide if you've done it correctly and you have the low power objective lens pointing down. Next, you're going to turn the lamp on and illuminate your specimen. Now remember, when it's on low power, what kind of light should you have? High intensity light or low intensity light? Low power, low light. So I've adjusted the lamp and it's on low power, low intensity illumination. Now, whenever using a microscope, if you wear glasses, you can take the glasses off because you don't need glasses when using a microscope because you are going to be focusing it for your eyes only. For your eyes only. That was a theme song to a James Bond movie many years ago. All right. Notice the arm. The arm should face you when you are using the microscope. Don't use a microscope this way, but rather use the microscope this way, so the arm is facing you. Good reason for it, by the way. The entire arm can tilt toward you, so that you can be sitting comfortably in your chair. You can arrange and position, almost like a recliner chair. You can position the arm of the microscope so that you can view it comfortably from where you are sitting. In fact, I am positioning it now comfortably. And oh boy, who remembers what eye you should be looking with into your eyepiece lens? Which eye, everybody? The dominant eye. So your dominant eye is looking through the microscope. Your non-dominant eye is open as well so that you can actually see in your book, and you can actually be drawing while you're looking back and forth with both eyes open so that you are not straining either of your eyes. All right, right now I'm looking into my microscope, and all I see is nothing but a big white circle. That's because I haven't focused it yet. So what you need to do, starting with the coarse adjustment knob while looking into the microscope, you're going to slowly move the coarse. Oh, wow. Whoa, cool. You're going to slowly move the coarse adjustment knob until you start seeing an image in focus, which I do now. And I only moved it a little bit, but I now see an image in focus. And I don't see all of it, however. I just see an edge. So what do? Well, right now I'm looking at it and the image is all the way at the bottom of my field of view. So I want to move the image up. How do I have to move the slide in order to move the image up? Say it out loud if you know. Hopefully you said the opposite direction, down, which is exactly what I'm going to do. So what I want to look at is towards the bottom, and I want the image to go up, so I'm going to move the slide down. Remember, the image is inverted. It is upside down and backwards from what it really is on the screen. So right now, I have the image totally fitting the center of my field of view. And it's a little bit out of focus, so I'm going to use the fine adjustment knob now to get it. Oh, now it's perfectly in focus. I can see it perfectly clear right now, again, with both eyes open. My dominant eye looking and my non-dominant eye free to look around and look at you. Why does it feel like somebody's watching you? Whee! Okay. Now, I'm currently looking at this specimen under low power magnification, but I know I've got to zoom in. I've got to go up to a higher power in order to see what's inside my specimen. So once you have it focused on low power, pay attention. We're going to use the nose piece. We're going to adjust the nose piece over to the medium power. When you do that, just to be safe, I'm going to watch from the side. But notice, I'm not turning the coarse adjustment knob anymore. Because once you have it in focus on low power, you should not have to move the coarse adjustment knob anymore. So it's in focus for me, even though I'm not wearing my glasses. I'm going to turn it over to the medium power. Plenty of room, plenty of room. And now when I look at underneath, oh, it's a little bit out of focus again. That's okay. That's okay. Don't use the coarse adjustment knob. Use the fine adjustment knob to turn again. Oh, there we go. And get the image in focus. Wow, everything is a lot bigger now. A lot bigger. I really zoomed in going from low to medium power. And now, in order to complete our lab and to make some observations for you to record, we're going to zoom into the high power. Now again, it's in focus on medium power. Do not touch the coarse adjustment knob. Use the nose piece lens. Watch from the side just to be sure and rotate. And when you do, wow, That high power objective lens is super duper close to the slide. It's just a hair off the slide. It needs to be that close in order for you to see it. But if you cranked up on the coarse adjustment knob and turned the high power over and then tried to use the coarse adjustment knob to get into focus again, smash, crack, shatter. Your glass would break. It's super close, yes, but you only need to use the fine adjustment knob. So when I look in, oh my goodness, it's pretty dark in there. How do you think we could brighten things up? Yeah, high power highlight. So I'm going to adjust the light a little bit brighter here. Oh, now I can see things, but it's a little out of focus. That's okay. Which knob should we use to focus our image? Yes, the fine adjustment knob only. So I'm going to adjust the fine adjustment knob so I don't shatter any slides. Oh my goodness. Wow, I had no idea the cells could look this big. Now, in order for us to complete the lab, we're going to have to move the stage around and hunt for cells that are showing us what we think we need to be looking for to tell the difference between a plant cell and an animal cell. But remember, the image is inverted, upside down and backwards. So while both eyes are open and while you're looking in, if you want to hunt the image to the left, which way should we move the stage? If we want to look to the left of the image, which way should we move the stage? To the right. That's right. So that's exactly what I'm going to do. Oh, wowee kazowie. If you want to go up, move it down. Oh, man. Before you settle into any one slide, you really want to hunt around and look for those cells that might represent those, oh boy, those things that you are really trying to find. So feel free to hunt around, move the slide around. I think I'm going to settle into this one here. Oh, no, wait, that one next to it is really, oh, wait a minute, there's an even better one. All right, one more. Oh, right there. Now, in your field of view, there's also a black needle. That black needle is connected to your eyepiece lens. And if you move the eyepiece lens around, it actually moves that little black needle around. So let's say you're looking for something in your slide. You can move and maneuver your stage so that that black needle is looking directly at whatever it is you are looking at, so that someone else, like your lab partner, can also look in there. And if necessary, use only the fine adjustment knob to focus. And that black needle will be pointing to whatever it is you wanted them to see. So use all of the tools available to you on the microscope to help you get your image into focus and to help you share what you're looking at with someone else. So I've taken you through the steps of how to view a slide, a specimen on the slide, how to use the microscope, and I hope you remember those steps. I'm now going to take you through the steps of how to shut down the microscope before I show you the images. I took pictures of these images that I'm going to share with you in a moment so that you can complete your lab activity. So, how do we pack things up at the end of our activity? Remember, slop it away. Pause me now so that someone can share with the class what you remember about slopping it away. Hopefully, you remember the acronym SLOP, which stands for Store Under Low Objective Power. Now, before we store these away, we want to do everything in reverse. So as we look at our microscope, we can actually crank down and turn off our light first. Turning the light off first will enable us not to cause a fire or allow the battery to run out or the bulb to run dead. Next, you can actually reposition the arm. Make sure the microscope is in the upright position, just like if you were on an airplane ready to land and you are asked to put your seat in the upright position. Please put your microscope in the upright position. Next, rotating the nose piece, return it to the low objective power. Please be sure to do that before you remove your slide from the stage, holding it only from the side and returning it into the proper location, typically a slotted box with the label of that particular slide on it. So now we have low objective power, body tube all the way down, lamp is off. We can now return the cover to the microscope and carry it back to the microscope cart or cabinet in the back of the lab, remembering your ABCs. And everybody tell me, what do they stand for? Everybody. Again. Okay, so at this time, I am going to end this particular video so that I can go to the next one, which will show you the magnified images that I recorded for you earlier so that you can actually complete your lab activity. So make sure your book is still open. You're going to record similarities and differences, hint, hint, between the two slides that I'm going to show you, and then write your conclusion. We can discuss it. But for now, I'm going to say bye-bye.